My dear students, welcome to CEDEES Institute, and today the topic of our discussion shall be about the urea cycle. Okay, right. Now, why is this cycle so important? What's the function? Urea cycle is the end product of protein metabolism. So all the amino acids which contain a lot of this nitrogen, is basically converted into ammonia, and unfortunately, ammonia beyond a certain limit is toxic. Ammonia is important for the formation of non-essential amino acids, for the formation of purines and pyrimidines, but beyond a certain limit, it's extremely toxic. I'll explain how and when the ammonia becomes toxic. So body has to find out a mechanism wherein it converts this ammonia into something which is easily can be flushed out of the system, and that is where your urea cycle comes in. It is converted into urea. Urea cycle basically takes place in the liver, and this urea is ultimately flushed out from your kidneys. Before I begin with the urea cycle, let us just see how exactly this urea structure is, right? So if you look at the urea structure, it has majorly, it has it has two amino groups. It has a carbon atom, right? So if you look at it very carefully, this is what your end product is. This is what urea is basically. It has two amino groups and it has a carbon atom. So let's keep that in mind. Now let's start. Every cell in the body produces ammonia, right? Because every cell contains amino acids. So let's start with your brain, or your nervous system. And now let us say that the ammonia that is basically formed there, ammonia that is formed in the brain, beyond a certain limit is extremely toxic. So what does the body do? As soon as the ammonia is basically formed, it is trapped by something that is called as glutamate. Okay, please capture these terms very well. Glutamate will trap this ammonia, right? So you have this ammonia, and with the help of an enzyme that is called as glutamine, I'll just write it down, glutamine synthetase. Okay, the enzyme is called glutamine synthetase. Glutamate is then converted to form something that is called as glutamine. Okay, glutamine. I would like to pause here. Glutamine is the most important storage and transport form of ammonia in the body. Which one? Glutamine. Okay. You also have alanine, which is a major transport form of ammonia from the muscles to the liver. But basically, let's say your brain, your major parts of the body, it's mainly your glutamine. As soon as the ammonia is basically, which is formed, it is trapped by glutamate, converted to glutamine with the help of glutamine synthetase, and this glutamine then becomes the major storage and transport form of ammonia. It's freely diffusible, it moves across cells, nothing to worry, glutamine. Okay. Now, why is this so toxic? My dear students, if you have some background about your Krebs cycle or your citric acid cycle, we have heard of something called as alpha ketoglutarate, right? So we have alpha Kg. This alpha ketoglutarate, if you realize in your Krebs cycle, alpha ketoglutarate, then you have succinyl-CoA, and then you have succinate, then you have fumarate, malate, it goes like that, right? So this alpha ketoglutarate combines with ammonia to form your glutamate. Let us say that this is taking place in your brain, your central nervous system, in your brain. So alpha ketoglutarate combines with ammonia with the help of an enzyme that is called as glutamate dehydrogenase. Okay, glutamate dehydrogenase gets converted to what is called as glutamate. Now, this is a particular reaction which can go either way. It's a two-way reaction. Now, when the ammonia in the brain increases too much, this reaction shifts to the right. Means what? There's too much of ammonia, then a lot of alpha ketoglutarate is used, and the whole thing is converted into glutamate. This glutamate will be converted to glutamine, and it just goes on. But what is happening here is in the brain, because of too much of ammonia, all this alpha ketoglutarate is now used up to convert it into glutamate. So what happens to alpha ketoglutarate? If there is no alpha ketoglutarate, your Krebs cycle will not take place. If the Krebs cycle does not take place, you don't have ATPs, and if you don't have ATPs, and that is where the toxicity of brain begins. Your slurriness and everything starts off from there. So that is how ammonia, even to a certain, even a little amount, if it becomes excess, it results in toxicity. Now, before to start with urea cycle, this particular glutamine, which contains the amino group here, which is trapped here, has to be released, right? So how is that released? Let's say it comes back to the liver, and here in the liver, I think I just write it down like this. This glutamine can be converted back to form your glutamate, This is basically a hydrolysis reaction. Water is added up. Basically a hydrolysis reaction. Glutamine is converted back to glutamate with the help of an enzyme that is called as glutaminase. Glutaminase. Okay, I think the board is getting a little bit crowded here, but I hope you understand this now. Now, once this glutamine is converted back to glutamate with the help of glutaminase, there is this ammonia is basically free in the liver to enter into your urea cycle. All right, let's learn these three reactions because there are three enzymes here. The first one, every cell in the body which produces ammonia is immediately trapped by glutamate. Glutamate traps the ammonia, converts it into glutamine. What is the enzyme there? Glutamine synthetase, right? Now, and this glutamine, when it comes back into the liver, can be reconverted back to glutamate with the help of an enzyme called glutaminase, right? And ammonia is liberated, which can then be used for your urea cycle. Why is ammonia so toxic to the brain? Whenever the ammonia is produced in excess in the brain, ammonia immediately combines with alpha-ketoglutarate and it is converted to form your glutamate. So whenever there is excess of ammonia produced, this reaction shifts to the right. Shifts to the right in the sense there is more and more of alpha-ketoglutarate and ammonia used and more of glutamate. So this reaction is shifted to the right. So the amount of alpha-ketoglutarate present in the brain decreases a lot. If there is no alpha-ketoglutarate, if you know that in your Krebs cycle cannot take place. If there is no Krebs cycle, there is no ATP. If there is no ATP, 70% of our brain utilizes so much of the food that we eat is used for the ATPs which are produced by the brain. So you don't have Krebs cycle, you don't have ATPs, your brain is just going to come down. Right, now let's start off with the urea cycle with this brief background as to what all is going backside. The first step, the first two steps, the urea cycle contains 5 important steps. The first 2 steps takes place in the mitochondria, and the next 3 steps basically takes place in your cytoplasm, right? So let's begin. The first step in urea cycle is carbon dioxide plus ammonia, okay, forms something called carbamoyl phosphate, okay, carbamoyl phosphate. And this takes place with the help of an enzyme that is called as CPS1. In other words, carbamoyl phosphate synthase 1, right? And I'll also write this in the bracket. Carbamoyl phosphate synthase 1 is allosterically activated with the help of something called as N-acetyl glutamate. But now, the first step, carbon dioxide combines with ammonia to form something called carbamoyl phosphate. This takes place with the help of an enzyme that is called as carbamoyl phosphate synthase 1, that is CPS1. What activates CPS1? N-acetyl glutamate. Just keep that in mind. Now, once carbamoyl phosphate is formed, carbamoyl phosphate then combines with something that is called as ornithine. And because ornithine is one of the first steps here, this urea cycle is also called as the ornithine cycle. It's also called as the Krebs-Henselate cycle. Okay, carbamoyl phosphate combines with ornithine to form something called as citrulline. Okay, yes, and this takes part with the help of an enzyme that is called as ornithine transcarbamylase. I'm hoping that you all have your textbooks along, because if you're not able to understand what I'm writing, the spelling, you can always look into your textbooks. So ornithine combines with carbamoyl phosphate to form something called citrulline. The enzyme here is called ornithine transcarbamylase, right? This step here, carbon dioxide plus ammonia utilizes 2 ATPs. 2 ATPs are consumed, right? Now, until citrulline, that is until here, this takes part in your mitochondria. And after that, citrulline that moves out from the mitochondria, it enters into your cytoplasm, and the rest of the steps basically takes part in your cytoplasm. Okay. Now, what happens next? Citrulline then combines with something that is called as aspartate, right? Aspartate, to form something that is called as arginosuccinate. Arginosuccinate. Okay, if this is forming arginosuccinate, that means the enzyme here to produce arginosuccinate is what is called as arginosuccinate synthase. Okay, arginosuccinate synthase. It's very easy to understand. So far we have learned 3 important enzymes. The first enzyme is carbamoyl phosphate synthase 1. The second enzyme here is ornithine transcarbamylase. The third enzyme here is arginosuccinate synthase. Now, What did we learn about urea structure? Urea has 2 amino groups, right? The first amino group comes from here, this guy. The second amino group comes from your aspartate, and this step also utilizes 2 ATPs. So far your urea cycle has used up 4 ATPs. It basically uses 4 ATPs. Okay. Next, arginosuccinate then is acted upon by an enzyme which is called as arginosuccinase to produce something that is called as arginine. Okay, arginine. I'm just going to write the enzyme here as like this, arginosuccinase. Now this step is very important because it results in the formation of something that is called as fumarate. Okay, all of you, I hope you're with me. Arginosuccinate is acted upon by arginosuccinase to produce arginine, and in this step, as arginine is formed, it forms your fumarate. Have you heard of fumarate? Yeah, it comes in your Krebs cycle, right? We have alpha-ketoglutarate, then succinyl-CoA, succinate, fumarate, malate, oxaloacetate. This is the fumarate which is seen in your Krebs cycle, right? Arginine. Then your arginine is acted upon by your last enzyme which is called as arginase. Okay, the fifth enzyme here, and it gives off urea and it gives off ornithine. There you go. This is the final production of urea. Urea is what you see here, 2 amino groups, 1 amino group to begin off here, the second amino group from the aspartate, the carbon atom is from the carbon dioxide which is seen here. This is in brief your urea cycle. Let's recap this once more, all of you, so that it just stays in your mind. Then you don't have to really look into your books. There are 5 main enzymes. The first 2 steps takes part in your mitochondria, and after that citrulline moves into the cytoplasm and everything else is then formed in your cytoplasm. The first step, carbon dioxide combines with ammonia to form carbamoyl phosphate. What is the enzyme there? Carbamoyl phosphate synthase 1. This CPS1 is different from CPS2 because CPS2 is used for pyrimidine synthesis. So pyrimidine pathway, we will not bother about it time being. The CPS1 is very much dependent on the allosteric activator called N-acetyl glutamate. So you have this there, carbamoyl phosphate. This carbamoyl phosphate combines with ornithine. If you realize, if you've done your Krebs cycle, this looks very similar, isn't it? You have your oxaloacetate which is formed at the last. This oxaloacetate then again combines with your acetyl-CoA, and then the whole Krebs cycle just goes on like that. So here, ornithine combines with carbamoyl phosphate to form citrulline with the help of an enzyme, ornithine transcarbamylase. This is the second. Citrulline is then acted upon by the enzyme called arginosuccinate synthase to produce something called arginosuccinate. Aspartate is added to this particular step here. Aspartate provides the second amino group for your urea. Arginosuccinate is acted upon by arginosuccinase to produce arginine, and this step basically gives off fumarate, and fumarate enters your Krebs cycle, right? Now this arginine is acted upon by the last enzyme called arginase to produce urea and ornithine in turn goes back into your urea cycle. It's as simple as that. Now there is an interaction. There is the Krebs cycle and the urea cycle. How do they come together? For example, the last step in your Krebs cycle is basically formation along with ATPs, your carbon dioxide and water in your Krebs cycle, right? That carbon dioxide is used for your urea cycle, right? Urea cycle requires 4 ATPs and Krebs cycle is used for producing your ATPs. That's the second way of interaction. The third and the most important is fumarate. This fumarate that is produced in your urea cycle, what happens here? It enters into your Krebs cycle, okay? This fumarate enters into Krebs cycle, and then what happens? Fumarate is converted into malate. Malate is converted into oxaloacetate, and once oxaloacetate is formed, my dear students, oxaloacetate undergoes transamination, and once oxaloacetate undergoes transamination, it comes back to form your aspartate. Aspartate can again re-enter your urea cycle. So it's this oxaloacetate can of course combine with acetyl-CoA, and it can go on in your citric acid cycle. At the same time, oxaloacetate can undergo transamination to form aspartate back again, and this can come back like this. So this is about your urea cycle, the first 2 steps in the mitochondria, rest in the cytoplasm, the 5 important enzymes, right? Next, there are certain diseases which come about when there is a lack of some of these enzymes. It's very, very easy to remember. Okay, first thing is we learn the enzymes. CPS1, ornithine transcarbamylase, arginosuccinate synthase, arginosuccinase, and you have your arginase. So if your body lacks CPS1, it results in what is called as hyperammonemia type 1. Okay, I'll just write this down, hyperammonemia type 1. Okay, if your body lacks ornithine transcarbamylase, then it is called hyperammonemia type 2. Please look into your books. Right, I'm just scribbling here. If your body lacks arginosuccinate synthase, you don't have arginosuccinate synthase, then you can think that probably is an excess of citrulline. So this results in what is called as citrullinemia. Citrullinemia, very, very easy. If your body lacks arginosuccinase, we don't have arginosuccinase. That means arginosuccinate is not acted upon. So then it results in what is called as arginosuccinic aciduria. I'll just write it down like this: arginosuccinic aciduria. Okay, and if you don't have arginase, that means there is an increase of arginine. Then it results in what is called as hyperargininemia. Hyperargininemia. Very, very easy. Let's repeat again. If you don't have CPS1, it results in hyperammonemia 1. If you don't have ornithine transcarbamylase, hyperammonemia type 2. You don't have arginosuccinate synthase, citrullinemia. You don't have arginosuccinase, it causes arginosuccinic aciduria. You don't have arginase, it causes hyperargininemia. I hope the urea cycle was pretty clear. The first thing to know is some of us get confused between what is glutamate, what is glutamine, what is alpha-ketone, what is all this doing with the urea cycle. With this small little background, these three enzymes, you should know that, right? One is glutamine synthetase, what is glutaminase, what is glutamate dehydrogenase, right? These three should be clear. And then of course your urea cycle with the 5 important enzymes. I think it's simple. You need 4 ATPs, the interaction between Krebs cycle and your urea cycle, and this particular small step, fumarate to oxaloacetate, and how it comes back to aspartate back again when it undergoes a deamination. I hope urea cycle is very clear. Just go through your books, go through each of the steps, just try to put it in mind, write it down, write down the enzymes, and I'm sure you don't have to worry about urea cycle beyond this. Thank you so much. Take care all of you. Bye-bye.